Hello, good afternoon. Today I'm going to talk about Enterococcus and Streptococcus ecorum and their role as early colonizers of the calf gastrointestinal tract. For the purpose of this presentation, early colonizers are the bacteria that arrive first to the gastrointestinal tract. So why do we focus on early colonizers? To answer this question of why are early colonizers relevant, first I want to explain the relevance of the microbiome itself in the context of a cow. So ruminants are efficient feed conversion machines because we feed them food that is unsuitable for human consumption, and they transform it into high-quality products like milk and meat for human consumption. They are able to do this conversion thanks to a very specialized microbiome that resides in the rumen. This microbiome is composed of fungi, archaea, bacteria, and protozoa. And all of these microbes work together to degrade feed particles, and in exchange of this degradation, they produce volatile fatty acids that the cow uses as the main source of energy, and then these microbes themselves serve as the main source of protein for the cow. But this very specialized microbiome that we found in adult cows, it's absent when the cow is born. The microbiome establishes through a process called microbial succession. In this process of microbial succession, the microbiome changes until it reaches a mature microbiome. At first, the bacteria that arrive are bacteria that are able to tolerate certain oxygen levels. They also have a wide substrate. They can utilize different substrates, and the ecosystem, thanks to these bacteria, start changing and allow the colonization by more specialized microbes, like, for example, cellulolytic bacteria, fungi, and protozoa, until we reach this specialized microbiome that can attach to feed particles and degrade it very efficiently. So these early colonizers prepared the ecological niche for further colonization. Besides helping in the microbial colonization, early colonizers are key also for the host development. We can see this in germ-free mice, where they have a reduced intestinal mobility, they have accumulation of mucus in the gastrointestinal tract, and they also have malformations in their lymphoid tissue. So if we go back to my first questions, why are early colonizers relevant? Well, they prepare an ecological niche for later colonizers, and they also interact with the host for an optimal gastrointestinal tract development. And why do we focus on these two taxonomic groups, Enterococcus and Streptococcus? Well, if we go to culture-based studies where they try to analyze the composition of early colonizers, we can see that both Enterococcus and Streptococcus are dominant taxa, and within the Enterococcus group, Enterococcus ecorum is the most frequent Enterococcus isolated in the first days of life. If we go to molecular studies where they use the 16S rRNA gene, that is a gene commonly used to classify bacteria taxonomically, we can see that in the first days of life, Firmicutes associated to Streptococcus dominate the microbiome. In this group, we also find the Enterococcus, both taxonomic groups. So that's why we focus in these two. And what did we do to study them? Well, we collected rectal swabs from calves in their first 3 days of life, and we used 13 calves that were part of a larger project. In this project, the calves received 3 different diets: colostrum, fresh colostrum, frozen colostrum, and milk replacer. So we collected the rectal swabs from these calves, and from the rectal swabs, we isolated bacteria. With the bacteria that we isolated, we culture them in the lab, and then we separate the cells, the bacteria cells, and the supernatant with all of the substances that the bacteria secretes. With the cells, we studied their capacity to produce biofilm, to utilize different substrates. We extracted DNA to classify them taxonomically based on the 16S rRNA gene, and then we also did whole genome sequencing of representatives of these groups. With the supernatant, we study the antimicrobial activity, the hemolytic activity, and also the production of volatile fatty acids. So based on the 16S rRNA gene, we collected 129 different bacterial isolates that belong to different genera, different taxonomic groups like Enterococcus streptococcus, Limosil-lactobacillus, Ligilactobacillus, Clostridium, and Paraclostridium. But for this presentation, we're going to focus only in Enterococcus and Streptococcus. That were, as I mentioned, and in line with the literature, the dominant taxonomic groups in the first days of life. We study how they utilize different substrates, so we group them with inulin, lactose, and maltose. Inulin, it's a prebiotic fiber that is commonly given to calves. Then lactose is present in milk, milk replacer, and colostrum. And maltose, once the cow starts eating solid, these bacteria will also be presented with maltose. As we can see, all of our isolates are able to utilize these 3 carbohydrates. And from the ecological perspective, this is important because we suspected that these microbes can utilize different substrates, and then they produce other products, simpler products, that other microbes can utilize, so this enables the colonization of other taxonomic groups. One of the products that these microbes can produce with the degradation of these carbohydrates is volatile fatty acids. The main volatile fatty acid that our isolates produce is lactic acid, especially the Streptococcus. Here we have the mean production, and we can see that the Streptococcus produce more than the mean, and most of the Enterococcus less than the mean, but regardless, both of them produce high amounts of lactic acid. Lactic acid has a dual role from the ecological perspective. It can act as it can be part of cross-feeding, where other microbes can utilize it, but it can also inhibit the growth of certain bacteria, and for example, some pathogenic bacteria like E. coli and Salmonella. Then we tested if our isolates had antimicrobial activity. So what we see in this plot, in the y-axis, we have the growth of of 2 pathogens, Escherichia coli and Salmonella typhimurium, and in the x-axis, we have the supernatant of our isolates. Here, in the first 2 bars, we have the pathogens growing without any inhibitory substance, and we have 2 different media, BHI and MRS. What we can see is that, especially for the dark bars, the MRS media, there is a reduction in the growth of the pathogens. But for the statistical analysis, I considered both media, and I highlighted in bold the isolates that, considering the effect of both media, have a significant reduction in the growth of the pathogen. We can see that 5 isolates are able to inhibit Escherichia coli, and 9 isolates are able to inhibit Salmonella typhimurium. As a side note, these 2 pathogens are common causal agents of diarrhea in calves. Then we wanted to test how safe these isolates are from the perspective of the host, so we collected erythrocytes from the cow and fed them to the supernatant of our isolates and measured the hemolysis ratio. These 3 dots here are 100% hemolysis, and these here are 0 hemolysis, and we can see that all of our isolates are below the 0 hemolysis, so we consider them non-hemolytic. Furthermore, we also studied their potential to produce biofilm. For the biofilm production, the y-axis here is the biofilm production. Here we have all of our isolates, and we can divide them in 4 categories based on these 3 cutoffs that are in the black lines, as non-biofilm formers, weak, moderate, or strong biofilm formers. Our isolates go from non-biofilm formers to strong biofilm formers. Why is biofilm important from the ecological perspective? Well, in the rumen, biofilm exists as a gelatinous matter that once they find a feed particle, it will attach, and it accelerates the degradation of the feed particles. And from the perspective of the bacteria, if they're able to produce biofilm, they have a higher chance of staying longer in the compartments of the gastrointestinal tract, so it's an ecological advantage for colonization. Okay, and back to the method slide. I already talked about all of these results. We're just missing the genome sequencing. So for the genome sequencing, here are our isolates From this table, I just want to show that our genomes after assembly were complete, they have low contamination, and our Enterococcus and Streptococcus belong to the species Enterococcus secorum and Streptococcus equinus. This is a phylogenetic tree of those genomes. In green, we have the Enterococcus, and in purple, the Streptococcus. And here we have the notation of biosynthetic gene clusters that are genes that have that encode for bioactive molecules that have the potential of having antimicrobial activity. So besides the lactic acid production, these genes could also explain the antimicrobial activity that we saw in the previous result against Salmonella and E. coli. Then, these two genus, Enterococcus and Streptococcus, have representatives that can work as pathogens in different contexts. And for example, Enterococcus secorum is a known pathogen in poultry, and we wanted to understand why, in the context of the cow, they don't seem to be causing harm, but actually helping in the development of the microbiome. So what are the differences between the pathogenic Enterococcus and the commensal or beneficial Enterococcus that we're having here? We're just starting with this analysis, so far what we did is we studied the single nucleotide polymorphism of the genomes of the pathogenic in orange and our beneficial or commensal in blue, and we see that are different from the pathogenic, but we still need to see which specific genes are the ones that differ. And now, well, the role of Enterococcus secorum and Streptococcus equinus as early colonizers seems to be involved with adapting the environment to allow the further colonization of other taxonomic groups by utilizing complex substrates and producing simpler substrates for their utilization, and also with biofilm formation. And they seem to benefit the host by preventing infection by the production of lactic acid or bioactive molecules, especially in this stage where the calf immune system is naive and they are able to outcompete these diarrhea pathogens. And we still need more research, especially to understand how they interact with the host and how they differ from pathogenic representatives of the same bacterial species. And with that, I'd like to thank you all for listening to my talk. Thank you to my lab, the Mantovani Lab, the Dairy Innovation Hub, and all my committee members.
Okay, the floor is open for questions for Fiorella.
Hi, Fiorella, nice talk. I have a question for you. Since your calves were enrolled in 3 different treatments, one that received frozen colostrum, the other received fresh colostrum, and the other one did not receive colostrum, did you see any differences between groups on the early colonizers that you got?
Yes, at the end, we were not able to gather enough data to study the difference per treatment, but an interesting observation that we saw based on the treatments is that the only Clostridium perfringens isolates came from cows that did not receive colostrum. So that's an interesting finding. They could be considered clinical isolates because these cows, as you remember, all the cows that were in the control group got sick. So yes, but we did not have enough isolation from the 3 treatment groups to be able to say these are the differences of isolates based on the treatments. You're welcome.
Other questions?
Very nice presentation, Fiorella.
Thank you.
Considering the importance of those species, is there a way to manipulate the rumen to increase their abundance?
Yes, so our lab, I think, is going to the pathway of trying to manipulate the rumen microbiome in the early stages of life, as as Arlen mentioned. The early stages of life is the best moment to modify the microbiome. So we think that if we feed a microbial cocktail, for example, in these stages of life, we can do changes in the rumen, because these strategies of changing the microbiome have been done with adult cows, and what we see is you can change the microbiome, but after you stop the treatment, the diet or the probiotic or whatever you're giving to the cow, the microbiome will bounce back to its original state. So we have the hypothesis that if we do this alteration at the beginning of life, we can see more long-lasting effects, but we still need way more research to actually confirm this to you. So yeah, we think we can feed them as a microbial probiotic in the diet.
So do we understand what it is that controls the microbiome in an individual calf?
Excuse me, can you repeat?
Do we know what it is that controls the microbiome? It's interesting that you can inoculate, but then it just goes right back to the way it was before.
Yeah, so, well, Professor Francisco has an article where they found that the genome has a huge weight on the microbiome, but also the environment. So if you're changing the microbiome, but you keep the cow in the same environment, once the alteration is removed, it will also bounce back. The ecosystem around it will help it to bounce back to its original state as well.
So Francisco, are we going to get this in the net merit equation right away so we can start selecting for this?
We should.
Soon. It's just right around the corner, I think. Good. Any other questions for Fiorella?
I wonder how heat stress might impact that.
Yeah.
Maybe this summer we get that answer.
Yes. Well, we see more cases of diarrhea in summer, so for sure there are microbial differences with heat stress.
All right, let's give Fiorella one more big round of applause. Thank you, Fiorella.